I'm John Strum, and this is Real Talk MS. It's March 3rd, and we have a lot to talk about. And it seems that, especially over the past couple of years, everyone is talking about GLP-1 receptor agonists. You know them by their trade names like Ozempic, Wegovy, Mounjaro, and Zepbound. And while they're best known as drugs that manage diabetes and promote weight loss, researchers are finding that these drugs are also effective in reducing the risk of heart attack, reducing high blood pressure, protecting against chronic kidney disease, improving fatty liver disease, reducing addictive behavior, and potentially mitigating cognitive decline. So what about MS? My guest this week is Dr. Ellen Mowry, the principal investigator in a clinical trial that's been funded by the International Progressive MS Alliance. And this trial is focused on determining whether a GLP-1 drug can reduce brain inflammation and protect neurons in people living with progressive MS. But before we get to my conversation with Dr. Mowry, there are a few other things that you should know about. We're just a few weeks away from Walk MS here in Los Angeles. I'm participating this year, and I could use your support. This podcast is all about people in the MS community who are making a difference, and today, you can be a difference maker. If you find value from listening to this podcast and it's something you can financially manage, I hope you'll take a minute to visit realtalkms.com/walkms and support me by making a donation in any amount. That's realtalkms.com/walkms, and you'll find that link in today's show notes. Real Talk MS is about communicating cutting-edge science in plain, easier-to-understand language. So before I tell you about something that I absolutely know you're going to want to hear about, we have to review some new vocabulary words and terms. The first word is proteome. The proteome encompasses all proteins in a living organism, tissue, or cell at a given time. The proteome is dynamic and will respond to diseases or environmental influences. The next word is proteomics, and proteomics is the study of the proteome. Mass spectrometry is an analytical technique that separates and measures atoms or molecules that carry an electrical charge in order to identify and quantify chemical substances or molecules. It's a fundamental technology in proteomics that enables the identification and quantification of thousands of proteins that are found in complex biological samples. And finally, the last term is protein panel. A protein panel is a collection of several proteins that are analyzed together as a biomarker pattern in order to detect and monitor certain diseases or to assess the course of a disease. And I'm introducing these terms because an interesting and important discovery has been reported from the Max Planck Institute of Biochemistry in Germany, where a research team used new mass spectrometry methods to analyze up to 2,000 different proteins in the cerebrospinal fluid of 5,000 people with a variety of neurological conditions, including MS. And the research team was able to identify a protein panel, a collection of proteins that, when analyzed together, serve as a reliable biomarker for MS, allowing for a confirmed MS diagnosis. Now, this in itself is good news. But there's even better news resulting from this research. By analyzing hundreds of MS patient samples, the researchers showed that the cerebrospinal fluid proteome at the time of diagnosis was associated with the level of disability years later. In addition, these protein patterns identified those patients who were going to transition from a relapsing to a progressive disease course more quickly than others. This suggests that important aspects of future disability and disease course are reflected in the cerebrospinal fluid proteome from the very beginning. In other words, this research shows that the biological information required to predict an individual's future MS disease course is already present at the time that individual is diagnosed. And that could be a game changer when it comes to diagnosing MS and determining the MS journey an individual is going to experience in the years ahead. We'll be sharing further developments as this cutting-edge science begins to make its way out of the lab. And in the meantime, if you'd like to review the details of this work, you'll find that link in today's show notes. In just a couple of minutes, we're going to talk with Dr. Ellen Mowry about a clinical trial that's being funded by the International Progressive MS Alliance. But as most of you well know, MS treatment isn't or shouldn't be limited to drug therapy. That's why the International Progressive MS Alliance has also launched a first-of-its-kind well-being research pipeline. And as part of that pipeline, the Alliance has awarded just over $8.1 million to fund 3 global studies aimed at finding solutions for the most common symptoms experienced by people living with progressive MS, including fatigue, cognitive impairment, pain, and mobility. One study is a comparison of self-guided, coached, and therapist-delivered pain self-management in adults with progressive MS. Led by Dr. Dawn Eady at the University of Washington School of Medicine, this study will compare a telehealth-focused therapy and wellness program that provides pain self-management strategies with a similar online program that can be completed independently or with support from a coach. From the study, Dr. Eady and her team will create and share several key resources, including data to support adoption in different settings, workbooks, global implementation plans, and more. Full disclosure, I serve on the University of Washington MS Rehabilitation and Wellness Research Center's Community Advisory Board, and I've participated in discussions in support of this project. The second study is being co-led by Dr. Lara Patouti at the University of Ottawa and Dr. Sarah Donkers at the University of Saskatchewan in Canada. And it's going to test whether priming the brain using transcranial direct current stimulation, aerobic exercise, or a combination of both prior to task-specific training for cognitive and mobility skills can improve outcomes. This study is expected to inform which combination of priming and rehabilitation provides the best functional improvements in mobility and cognition for people living with progressive MS. And the third study being funded is a multimodal, tailored, and adaptive exercise program to improve walking capacity in people with progressive MS and abnormal fatigability. Led by Dr. Peter Freys at Hasselt University in Belgium, this study will examine whether a specially designed exercise program can help someone with progressive MS walk farther and more safely in their daily lives by reducing their symptoms of fatigability. That's the decrease in function someone experiences the longer they walk. Participants in the study will include people with moderate to severe disability, which is an often overlooked group, and each participant will undergo a personalized exercise program tailored to address their specific walking challenges, and that exercise program increases in intensity over time. If proven effective, this intervention could become a new evidence-based approach to improving mobility in real-world settings. Like everything the International Progressive MS Alliance does, each of the research teams for these studies consulted an advisory board or engagement team of people with progressive MS to provide input on the study design and requirements, and the researchers incorporated that feedback into their proposals. In addition, the Alliance's People Affected by MS Engagement Coordination Team participated in reviewing the proposals to help decide which would be selected for funding. We'll be following this work and sharing further news as it's announced. In the meantime, if you'd like to learn more about the International Progressive MS Alliance, you can visit their website at progressivemsalliance.org, and you'll find that link in today's show notes. Drug manufacturer Merck has teamed up with the Mayo Clinic to launch a research collaboration that will use artificial intelligence and so-called big data to speed up drug discovery and development. And the initial focus of this collaborative effort will be diseases with high unmet needs, including multiple sclerosis. The Mayo Clinic is bringing their extensive clinical and genomic databases to this collaboration, and Merck is bringing their AI-enabled research technologies, including their virtual cell platforms. These are computer-based models that simulate how cells act and react in healthy settings and in certain disease settings. The goal of the collaboration is to improve how drug targets are identified, and it's the hope that this will increase the likelihood that experimental therapies will ultimately succeed. The Mayo Clinic's datasets include laboratory results, medical imaging, clinical notes, and molecular and genomic data, all of which will be de-identified. That is, everything will be 100% anonymous. And this effort should produce a more accurate model of diseases, which should lead researchers to better predict which drug targets are likely to succeed. Initially, this collaboration will focus on MS, inflammatory bowel disease, and atopic dermatitis. And it represents one more example of how AI is impacting healthcare, from the laboratory bench to the bedside. We'll keep you updated as this work gets underway. GLP-1 receptor agonists may be the one class of medications that almost everyone is familiar with. We just know them by their more popular trade names like Ozempic, Wegovy, Mounjaro, and Zepbound. And they could be considered the miracle drugs of the 21st century. But could they reduce inflammation in the central nervous system and offer neuroprotection for people living with progressive MS? Well, that's the question my guest, Dr. Ellen Mowry, is hoping to answer. And in a moment, we'll meet Dr. Mowry. GLP-1 receptor agonists are a class of medications that have become very well known by their brand names, names like Ozempic, Wegovy, Mounjaro, and Zepbound. These popular drugs are used to manage diabetes and to promote weight loss. They've also been shown to reduce the risk of heart attack, reduce blood pressure, protect against chronic kidney disease, improve fatty liver disease, reduce addictive behavior, specifically addiction to alcohol or opioids, and potentially mitigate cognitive decline. But what about MS? Well, last December, the International Progressive MS Alliance announced that it was funding a 2-year clinical trial to investigate whether GLP-1 receptor agonists can reduce brain inflammation and protect neurons in people living with progressive MS. Joining me today to discuss this work is the study's principal investigator, Dr. Ellen Mowry. Welcome back to the podcast, Dr. Mowry.
Don, it's always a pleasure to be here with you.
GLP-1s like Ozempic or Mounjaro are currently household names for weight loss and diabetes. What was the aha moment that led you and your team to believe these drugs could specifically protect neurons in progressive MS?
It's a great question, John. We've talked before about how, we've been on a journey to figure out what are the things that contribute to different outcomes for people with multiple sclerosis. Why do some people develop progressive symptoms later in the disease course while others seemingly are quite stable? And as you know, we've done a lot of work in the past looking at cardiometabolic disorders. So that's a big mouthful, but I like to think of things like diabetes, hypertension, hyperlipidemia, obesity. And, this isn't just work that's come out of our lab. Many labs have tried to look at this and demonstrated that outcomes generally are worse for people with MS who are living with these comorbidities. And we and several other people have tried to look at interventions that might help out to reduce some of these comorbidities through dietary modification or exercise programs and things like that. And they sometimes show some good short-term promise, but these are very hard things to maintain. And so over the past several years, as we've been thinking, how are we going to actually sort of work on these chronic illnesses, and as it sort of becomes more and more clear that these are highly genetically driven comorbidities that require treatment, just like any illness does, right? These aren't things that can just sort of be done by will alone. We really started to get excited about the prospect of using medications known to work for those conditions, like GLP-1 receptor agonists. And then, of course, my collaborator, Dr. Peter Calabresi and Dr. Marjan Garagoslu in the lab were demonstrating that in mouse models of MS, the GLP-1 receptor agonists that we're using in the trial did protect nerve cells from dying. And nerve cell death is what actually contributes to progressive MS symptoms. So that was very exciting. And there's a lot of excitement in general about this class of medications in other illnesses in which nerve cells die, things like Parkinson's disease and Alzheimer's disease. So, I think the field is generally considering that these medications, whether by mitigating these concomitant illnesses or by having direct effects on inflammation going on inside the brain that leads to nerve cell death, may have a role for protecting many people in preventing disease from starting or slowing down disease that has begun.
The GLP-1 receptor agonist that you'll be using in this trial is NLY-01. How does NLY-01 differ from the GLP-1s people might be taking for other conditions?
NLY-01 is provided by Neurally, the company that's partnered with us to provide medication for the trial. And essentially, it's a pegylated version of an approved GLP-1 receptor agonist called exenatide. And the pegylation allows it to kind of be longer lasting in the body. It's been demonstrated in animal models to cross into the across the blood-brain barrier and get into the brain. So, if it does work and it's dependent on mechanisms directly in the brain, that's pretty reassuring. But they all, work very similarly. Our bodies make GLP-1 naturally, and it degrades very quickly. These synthetic agents are all designed to be degraded less quickly. And some of the FDA-approved products have other hormones or other kind of receptors that they target in addition to the GLP-1 receptor. But nonetheless, they're all hopefully designed to give a little bit more long-lasting effects compared to the one that's already synthesized in our body.
We often hear about the inflammation that happens behind the blood-brain barrier, even when there are no new lesions. How does NLY-01 specifically target the microglia to put out this inflammatory fire?
Yeah, that's a great question. In the mouse models of MS, our team demonstrated, in some nice experiments that it actually acted on the astrocytes, which are the star process glia, right? So the glia, we always think of sort of the support cells of the brain. They have a lot of function and impact. And in people with MS, it's thought that both activated microglia and toxic astrocytes have a role in promoting neurodegeneration. And so in the mouse models, they saw that NLY-01 reduced the activation of those astrocytes into the toxic phenotype, and subsequently led to less nerve cell death in the retina.
Most current MS drugs focus on the peripheral immune system. Can you explain how this trial sort of shifts the focus toward neuroprotection, actually saving the nerve cells that are already under inflammatory stress?
Absolutely. We are so fortunate in the field to have so many nice medications that are already available that prevent the nasty effects of the peripheral immune system. When we say peripheral, we mean the immune system that's out in the rest of the body. And in the case of people with MS, that peripheral immune system gets activated, gets into the brain and spinal cord, and causes damage. And those damaging effects are realized by the lesions or white spots that accrue in those areas that sometimes lead to relapses. And the medications we have already work to prevent those peripheral immune invaders from coming in and causing white spots and relapses. We're pretty good at stopping those activities from happening. What's the best way to do it for each individual person, et cetera, et cetera. It's still information that's being worked out, but we can get to the point where people aren't having relapses or new MS lesions. So I would consider that a person controlled from that perspective, yet we know that these other processes have been set in motion, like the glia cells of the brain causing ongoing inflammation in the brain and leading to increased and further neurodegeneration. And so I think a lot of work in the field is actually trying to figure out, how do you make repair? How do you optimize repair of myelin that's been damaged? How do you protect the nerve cells that are still there and give them the best shot of lasting as long as they can? Because as we get older, of course, the nerve cells die off for everyone, even if we don't live with MS. But it's accelerated in people with MS. It's accelerated in people with these concomitant conditions. So we're really all trying to figure out how do we slow down that process, because once enough nerve cells have died, people start to accumulate disability. And that is what we call progressive MS.
This trial utilizes some advanced MRI techniques. What are you looking for through these techniques that maybe a standard MRI could miss? And how will that tell us if the drug is working?
Yeah, so the main outcome for this trial is a more traditional MRI metric, looking at brain volume, which is a rough surrogate for how much nerve cell damage is going on in the brain. But at the same time, we wanted to learn not only a little bit about how the medication is working, if it does, but also to be sure that regardless of the results, there would be some new information that might push the field forward to help design the next study or think about what are the next medications to try. So we are looking at some special fancy MRI measures of, for example, metabolism in the brain. Also, we've added some of the newer metrics that were recently incorporated into the diagnostic criteria for MS. So looking at iron, deposition and things like that. And also trying to look at some newer measures using diffusion techniques, they're called, to look at the integrity of nerve cells. So I think all of those things will help us to better gain an understanding of what's going on in MS progression largely, maybe identify some additional treatment targets, but also, if the treatment is successful, to help us to understand potential mechanisms by which that's occurring.
Progression is difficult to measure. So I'm wondering what criteria are being used to identify participants for this trial?
Yes, as you know, John, the nomenclature for progressive, relapsing, and all the different types of MS has really gotten a bit blurred over time. And in the field in general, we generally like to think of the idea that we know nerve cells are actually dying even early in the course of multiple sclerosis. So there isn't a magic time point at which, you were experiencing only relapsing MS, and now you're only experiencing progression. It's sort of a continuum with a great deal of overlap. And in that context, we aren't interested in this trial in adjudicating whether these medications prevent relapses. So we are including people who seem to be stable from the perspective of new relapses and new spots on their MRI scan. But we aren't excluding people who don't appear to be progressing because of this notion that we know that nerve cell damage is accumulating under the surface in most people with MS. So that is another reason that we're using MRI scan as our primary outcome, because we can measure that nerve cell death even if people aren't noticing worsening yet on their clinical exams. And the idea, I think, should be for the field to identify people at risk of clinical progression where their symptoms are getting worse before that actually happens. And those are the folks we do want to include in these trials, because we want to see, can the medication stop it from happening in the first place?
We've had previous conversations about some of the extensive research you've done focusing on vitamin D and even diet and their influence on MS. Do you see this GLP-1 trial representing kind of a new chapter where we're using the body's own metabolic pathways to fight the disease?
I think that it is using a new tool in the toolkit that appears to be more powerful than, behavioral modification alone, for example, in the context of tackling, metabolism changes that might make MS worse. And when new tools become available in the toolkit and they look promising, we should try them, right? We know these medications have broad benefits for many people who have indications to use them that reduce the burden of many bad health outcomes that are related to sort of the toxic metabolism associated with diabetes or, carrying extra weight or whatever. And I just think we need to like help people improve their outcomes with whatever tools will work. And who knows, right? That's why we do the trial, if it will work. But we can't stop trying or throw up our hands and say, well, hopefully something new comes along. And I love the idea of identifying products that are already out there and saying, like, wait, if there's a rationale to try it, what are we waiting for?
Good science is never a short pathway. As I mentioned earlier, this is a 2-year trial funded by the International Progressive MS Alliance. And if the results are positive, what's the realistic roadmap for this becoming an available treatment for people with progressive MS?
I think that's a great question. And part of it may depend on payers and other things that are a little bit out of our hands. Traditionally, Phase 2 trials like this one are really designed to prove that there seems to be a signal there, but they are a little bit shorter and they have fewer people in them than larger studies. So often, especially if you're planning to go for an approval for that indication, like should people with MS just get GLP-1 receptor agonists, you do need to do a much larger study, right? Because you want to make sure that the results you're finding with the MRI scans translate to a clinical benefit, in terms of preventing real clinical disability worsening. The larger study also means that if there are unusual safety features or problems associated with the medication that don't come out in a smaller, shorter study, it's more likely that they will come out in the larger study. But the reason that we kind of started, and one other thing I would say actually is that in this study, we're including people with a body mass index of 27 and up. And, you're always kind of trading off the pros and cons of restricting versus widening who gets to be in your trial. Of course, people with lower body mass indices might have weight loss as a not needed consequence of taking a medication like this. But if the medication is working by sort of downregulating those toxic glia cells in the brain, it may be that like additional studies and people of lower weights actually do need to be done to figure out, well, for example, some of the GLP-1s have less weight loss associated with them than others. So maybe those would be appropriate for people who don't have as much weight to lose. And some of the others might be great for people who do have more weight concerns or, diabetes or things like that. So there are a lot of different holes that will need to kind of be filled in. We had chosen that, though, because at this time, some of the FDA approvals for GLP-1 receptor agonists occur if your body mass index is 27 or up, plus you have a relevant comorbidity. And my thought was, maybe MS is a relevant comorbidity, right? And maybe being strategic about the way we include people, not to say only people who have much higher BMI, but, backing it down to that slightly lower version might help us if it's a successful, pathway to get the medication more easily into the hands of the people who need it.
Well, Dr. Ellen Mowry, thank you for walking us through what sounds like an exciting trial. Thank you for all you do in the clinic and the lab to improve the lives of people living with MS. And thanks so much for talking with me today.
Thank you, John. And as you know, it's a team effort that includes all the different constituents, all the people who are really dedicated to making a difference and to finding answers for the people we serve, including and most importantly, those people themselves who are so generous in contributing their ideas and time and willing to participate in research.
That's going to wrap up this episode of Real Talk MS. Real Talk MS is powered by the National MS Society, and you can share this episode of the podcast by letting your friends or family members know that all they have to do is point their web browser at realtalkms.com/444. You'll find that link in today's show notes, so you can easily copy and paste it right into an email or a text. We're just a week away from MS Awareness Week. And this year, MS Awareness Week is focusing on the unseen aspects of MS. I hope you'll join me next week to hear firsthand how those invisible symptoms changed everything in someone's life. I'm John Strum. Thanks for listening. Stay safe and make healthy choices.